Alrighty, so we've spoken bare bones anatomy and physiology now, so let's just bring it all together and talk about what EEG actually is. So where does the signal come from? What are we actually measuring when we put the electrode on someone's scalp and get data that we can interpret as being neural activity? So let's just start by discussing what we're not measuring. We are not measuring presynaptic activity. In other words, anything to do with a neuron's actual output, like spiking activity or action potentials, we are not measuring that. These are too small, too brief, and they can't add together. Now, the deflection that we get from an action potential is quite tiny in the scope of the whole brain. They're also really quick. We're talking order of 5 milliseconds and it's over, and these aren't adding together. What this means is that neuron A here might fire an action potential, neuron B here might fire an action potential, but the potentials aren't going to add up. Like, they're just individual cells doing their own thing. However, we spoke about temporal and spatial summation in the last video, in which postsynaptic potentials can add together if they're occurring at the same time, but at different locations, or in the same location, but at different times. These also are a lot slower. So let's just revisit that concept now. So here we've got an excitatory synapse where this is the presynaptic neuron, so the axon terminal. We've had an action potential travel down the axon, we're at the terminal now. Calcium channels have opened, and this has led to the exocytosis of neurotransmitter. And in this case, it'll be glutamate. So in this example, glutamate will go across the synapse and it will bind to the dendrites of the postsynaptic neuron, which is here. If you remember, we've got these ligand-gated channels that sit on the dendrites. This will bind, these channels open, sodium will flow into the cell. This is going to lead to the membrane potential becoming more positive, meaning that this neuron is closer to its threshold, more likely to engage in an action potential. So inside of the cell, we've got a depolarization. So, you get this large depolarization at the dendrites, which we can see here. But let's have a think about what this is actually doing in the extracellular space. Because if we're having sodium flow into the cell, that sodium needs to come from somewhere, and it's coming from the space around the neurons. Now, if positive ions flow into the cell, this means that a positive ion is being taken from the extracellular space. Now, what this leads to is that we can measure now this sink in the extracellular space. So all the sodium is flown in, we're more negative on the outside now. Now, these ions that flow into the cell do need to leave the cell eventually, and they'll do this about at the cell body. So what we're going to get here is a slight positive deflection that we call a source. So we have a sink because the sodium has gone into the cell and left the extracellular space more negative, and then a slight positive source because those positive ions leave the cell eventually. The same, but inverse is true for an inhibitory synapse. So in this case, we've got a presynaptic neuron. GABA has now been exocytosed into the synapse. This is going to go across the synapse, bind to the dendrites of the postsynaptic cell. It will bind to the chloride channels on the ligand-gated chloride channels on the dendrites. These channels open, chloride will go into the cell. This will hyperpolarize the cell and bring this cell away from its threshold. It is now less likely to engage in an action potential. So same thing applies here. Well, we've got this hyperpolarization happening inside of the cell, but if chloride is leaving the extracellular space to enter the cell, that means that our extracellular space is now more positive, and we get a source. Remembering that this chloride has entered the cell, it will need to leave eventually. It'll do so at about here, and we get a negative sink around the cell body. What we now have is what we call a dipole. Now, a dipole is just a pair of positive and negative charges that are separated by a distance. Now, electrical dipoles are formed based on this idea of the extracellular space changing to either negative or positive depending on the PSP that has arrived at the synapse. So if we have a look, so one thing to keep in mind is that orientation matters here. So if we look here, we've got all of these negative ends of the dipole facing upwards. So this will have like a negative sum. However, if we've got this inverse relationship here where we've got negative, positive, what we're going to get is a cancellation of the charge. So this is now a net neutral, essentially. More realistically, we have neurons haphazardly arranged, everywhere in the cortex. It's hard to know exactly what we'd be getting at here. Could it be a net positive, a net negative, or a zero? Who knows? more dilemmas to consider with EEG. Firstly, the dipole from an individual cell is actually going to be really small. So it's not really too much good to us because, again, we're trying to record something from outside of the head. We need something a bit stronger. So an individual dipole, not going to cut it. The other thing is that obviously we have a thick layer of skull in the way. Now that's not the only layer. We've got skin, we've got hair, we have electrode gel, etc., etc. So let's have a think about what is it that actually, despite all these dilemmas, allows us to actually record something. Well, we spoke about the cortical layers in the first video, of which we have pyramidal cells that are arranged in a particular manner, depending on the layers of the cortex here. One thing to keep in mind, we don't really need to be too concerned about like the specifics of the layers, but you can see in this diagram, we spoke about this earlier, these pyramidal cells in the cortex are arranged in a parallel manner. So if they are parallel, then that kind of gets rid of that issue of the cancelling out of sums. The other thing is that we can get summation of these polarities because remember they are graded potentials. So if the neurons are synchronously active, then we're going to get a summation of the dipoles. So the cortex is really primed to be able to, allow us to record this because we have these dendrites that are arranged next to each other. And remembering we have these cortical columns that seem to operate as a unit. So if the neurons are all active, well, we can get summation here. Now we're getting something that's a bit more realistically measured from outside of the scalp. So let's have a think about how it gets from the brain to the electrode. So if that there sums up the actual generation of the signal within the brain. It's the dipoles that result from the change in the extracellular fluid based on the postsynaptic potentials. So volume conduction is how that positive and negative ions of, depending on, if it's an inhibitory or an excitatory PSP, we have these ions flow through the brain before they get stopped by an insulator. So what we have, volume conduction as a whole it's an electric current passing through biological matter from a source to a sensor. The source in our case is the brain, the sensor is the electrode. So this wave of charge travels through the brain, but then it gets stopped because we have a scalp, we have dura, we have CSF. Now CSF actually conducts electricity quite well because there's ions in there, but then we have skin, we have gel, and then we have the electrode. So what we get here is after the volume conduction has occurred, we stop engaging in volume conduction because it's been stopped, and it's now capacitive. What this means is that insulation essentially prevents those charges from mingling and neutralizing. So let's have a look at the sequence of events here. Firstly, let's say an IPSP has arrived at a synapse. Because that's an IPSP and chloride has gone into the cell, for example, the extracellular fluid is now more positive. That positive charge propagates via volume conduction, so it'll flow upwards until it gets stopped. What happens here is that because we have positive charges building up on this side of the insulator, we know that opposites attract. So if there's positive charges here, it's going to attract the negative charges present in the next layer. This, in turn, will then repel the negative charges. So we get positive building up here. The positive will attract the negative, so on and so forth. That will happen at each layer until we get to the electrode. Now, you might wonder, why do we actually need gel when we're recording EEG? And the answer to that is because air is a really shit conductor of electricity. So the gel is essentially thickened salt water. That allows these ions to actually travel in this arrangement until it reaches the electrode. However, slight caveat, now this will be discussed in more depth in future sessions, but I'll just prime it now. When we're measuring EEG, we're not measuring like the underlying cortex necessarily. The voltage that's recorded at each electrode is computed with a reference to something else. Now, there are lots of different referencing systems that you might engage in. So one that we commonly use is the average reference. What this means is that the voltage at one electrode is measured in reference to the average of all the other electrodes. We can also, reference to an electrically inert site. Now, a popular one is the mastoid, so the bone sitting just behind your ear. While this has been shown now to not necessarily be inert, it is a popular option. There are others. I'll leave that there now. We'll discuss that in more depth in future times. So, long story short on this one, EEG reflects the summation of active dipoles in the extracellular fluid from cortical pyramidal neurons that are, 1, present in large numbers, 2, parallel to each other, and 3, active at the same time. However, if we could stop there, like, this would be fantastic, but unfortunately that's not the case, because EEG also measures a lot of other shit too. That's going to be the focus of the next couple of weeks of content, so stay tuned for that one. Thanks for listening.